everyone, and thank you for staying and joining us for this panel session. It's my pleasure to welcome everyone. And today we're going to be talking about disruptive technologies in laboratory medicine. And having been in laboratory medicine for 40 years, when I think about disruptive technologies, I kind of related to my grandmother, who was born in 1897 and went through the years where, when she was young, they lived in a house with no running water, no plumbing, no electric, and rode in a horse and buggy. But yet, during the course of her lifetime, she lived to see space travel and a man walk on the moon. So when I think about those years, I think about, boy, that's a lot of disruptive technologies that occurred. But when I look back over the last 40 years in laboratory medicine, I think it pretty much equates to what went on during that time period. So when I started in the laboratory, and I've been a medical director for this many years, strictly in infectious diseases, we went from culture plates. The most radical thing that came along was an automated blood culture system, then automated ID and AST, going on to MALDI-TOF for organism identification. So that for us was disruptive technologies, but I think where we are today and the hugest field that has really revolutionized what we do in infectious disease in particular is in molecular diagnostics. So 20 years ago when my lab started doing molecular testing, it was all homebrewed PCRs, different rooms and days to get results. And now when we think where we are now, we're going to talk today about some technologies that the thermocycling is done in a little cartridge that's attached to your cell phone, and you can get the results in 20 minutes. So that's really revolutionized how we approach it in a lot of different ways, not only from the ability to have much more comprehensive diagnostics, but I think the key point now is that it's going from the laboratory setting to basically a point of care setting or near patient where the results need to be there and need to be very, very quick. So we're going to be discussing this today. Our panelists are here from a variety of different companies that have somewhat new and innovative technologies that we're going to talk about. And also our final panelist, James, say your name right, Boiani.
Perfect.
He represents the Coalition of CLIA Waiver Reform. And I think that's going to be really critically important as we move towards point of care and more CLIA wave testing in our particular areas. I'll have all the panelists introduce themselves, their companies and their technology, and what they feel is their positioning, not only in today's market, but where we're going to be 10 years down the road. I'd like them to think about that as testing becomes more decentralized. We moved to centralized laboratories because that was the only place where we could do this type of technology, and now it's really becoming much more decentralized back near the patients. And then afterwards, we're going to talk about specific questions, not only relating to their own technologies, but how they see it'll be integrated into laboratory diagnostics and the new challenges that we face as we move towards more CLIA wave point of care testing. Everything is very mobile now. Everything is technology driven, and there's a lot of bioinformatics. So there's issues like PHI and other things. So these are some of the topics that we'll discuss afterwards. So I'd like to start first with Kevin, and he could introduce himself, and you can talk a little bit about your company.
Excellent. Thanks, Christine. That was a really great introduction to this session, and proud to be part of it. I've been with a company called Quanterix for about 3 years now. It was founded about 10 years ago. The founder of Illumina with some ex-executives from Roche got together and they felt that they could digitize biomarkers in the blood and be able to see proteins much cleaner. And so they set out on a quest, and we actually teamed up with bioMérieux, I think about 5 years ago, to bring that technology in a fairly significant large platform into an IVD setting. And $150 million later, we invested and launched yesterday a little benchtop. And so the concept of getting to a point of care is clearly in our sights, and we're working with a lot of point of care diagnostic channels to market around trying to bring this technology there. But in the meantime, we've repurposed the larger instrument into pharma biotech, and just about every pharmaceutical company and biotech now owns at least one machine. Some of them own as many as 4 or 5 machines. And they're stratifying patient cohorts, and they're also monitoring biomarkers in the blood for their targets, as well as any of the off-targets that they want to make sure they're not perturbing, like many of the immune systems. So we can digitize 50 different cytokines, interleukins, interferons, and we can see them with incredible sensitivity. It's almost equivalent, we did the calculations, to a grain of sand in 2,000 Olympic-sized swimming pools, what our technology can see from a sensitivity standpoint. And from a specificity standpoint, we can see a single blade of grass in a field the size of Alaska. So it's taken rocket science and applied it to the blood, and we can see single femtograms per ml. And that has led then to being able to see disease long before symptoms. And so most of the pharmas now have them in their trials, phase 1s, phase 2s, looking at cohort patient stratification, as well as monitoring the performance of the drug. Someday we think there'll be a lot of companion diagnostics that's going to lead from that, and that's a big piece of our future focus. So it's exciting to be here, and I think the point of care is a real important topic for the future.
Yeah.
Thank you. I'm Neil Polwart. I founded a company called Novarum DX, which is now part of the BBI group. BBI specializes in developing lateral flow technology, providing reagents for diagnostic companies. And Novarum brings mobile technology to that suite. So we enable people to read and share test results from point-of-care tests, whether it's lateral flow or a different platform, and get that data back to wherever they need it to go. And we look at that from a kind of, I would say, a holistic point of view. So we're looking at that from the point of view of patient safety and how can we use the mobile device to enhance the patient safety rather than simply capture the results. So removing ambiguity, guiding the user through performing the test, doing what we call test choreography, so stepping them through all the steps of the process there, and enabling patients to do testing at home for themselves or enabling testing in settings which just weren't possible before, so in remote or rural areas.
Alidad, Mireskandari. I work for Interpace Diagnostics. Our company is a medical diagnostic company that develops tests for detection of cancers in GI and endocrine, specifically thyroid. What we've done with our technology is look beyond just diagnosing cancer. Thyroid cancer has a 5% mortality rate, but the incidence of thyroid cancer detection have been going up incrementally over the past 15 years or so. So we focused our energies on not only developing assays that accurately detect thyroid cancer, but also look at prognostication and identifying the patients who will progress to the more aggressive forms of cancer. We combine a number of different technology platforms, including next-gen sequencing that really looks at both mutations and RNA fusions with loss of heterozygosity and some other proprietary markers we look at, to really be able to take diagnosis of what is an indolent cancer and looking at ways to identify the subset of patients that will progress to a more lethal form of malignancy that can metastasize into their lymphs and other tissues and cause death.
Hi, I'm Dan Heller. The company is Two Pore Guys, which is an unforgettable name. We do solid-state nanopores, and solid-state meaning it's silicon, not a biological pore like Oxford Nanopore, and we do not do sequencing. We are a single-molecule sensor, and it's a very low-cost sensor, and the consumable, it basically is a glucometer-like workflow. So our device, reader device, costs about $100, and the consumables cost about $1, and it's a development platform. So because it's a single molecule sensor, and because we can discriminate targets from background, very little sample prep is necessary. The platform allows existing diagnostic reagents to be used in our platform, sometimes as is, sometimes with some modifications. Because of its single molecule sensitivity, we have pretty low limits of detection and other characterizations that are really very, not really possible in a, even in many cases, a desktop system. So we don't make assays ourselves, but as I said, we adopt others, and we have about 30 assays so far, ranging from cancer, liquid biopsy tests, infectious diseases, and agriculture. And We'll be talking later today about the technology and the UCSF study that we recently completed, testing cell-free cancer DNA in cancer patients at UCSF.
Hi, I'm James Boiani. I'm a partner at the law firm of Epstein, Becker, and Green, who focuses on FDA regulatory issues, and also the founder and general counsel of the Coalition for CLIA Waiver Reform. And so I'll try to keep my comments fairly brief. It'll be something between a fireside chat and a Twitter. Or tweet, sorry. So I think I was thinking about the way to discuss the issue most easily and approachably. And I was thinking probably just give me a brief background on my own experience. Prior to becoming a lawyer, I was a chemist, and I worked as a consultant to the Environmental Protection Agency on something called the Alternate Test Procedure Program. And that program was designed to allow diagnostics manufacturers to bring tests to compliance monitoring for water quality. And the idea was assessing whether those new technologies and new tests were comparable to the old tests that were used to establish the guidelines that people needed to comply with. Did that for several years and really sort of built my interest and experience in assessing method comparability. That's all the program was. I went to law school, got into FDA issues, started doing a lot of pharmaceutical work for several years. On drug approvals and all of that. And then someone came into my office one day and said, oh, I've got a diagnostic test. I'm like, oh, I know about diagnostic tests. Yeah, I'm having trouble getting a CLIA waiver. I didn't know what that was. This was about 2008. And so I start researching CLIA waivers, look at the statute, look at the legislative history, all the legal stuff. And it looks to me like, okay, a CLIA waiver is pretty straightforward. What you need to assess is whether the test is simple to use, which would be like with a human factors or usability study. And you assess whether it's accurate. And accurate for CLIA waiver purposes means that untrained users, that would be physicians, lab techs, nurses who are using the product in a CLIA waived environment, get the comparable results to the experienced lab techs who can run the test. So I said, oh, that seems pretty straightforward. You run a method comparability assessment, you do an agreement study essentially, and you can show that the user's not affecting the results. Great, you go get your waiver. Well, it wasn't that easy. Over a 3.5-year ultimately appeal process, we kept getting lots of feedback of, well, that's not how CLIA waiver works. You need to evaluate total error. And I was like, well, it's about the user. It's not about the technology. This product's already 510K cleared. You've said the sensitivity and specificity is okay or acceptable for the intended use. That establishes the benchmark. Let's go with that. No, you need that. Oh, you need a reference method. Oh, and basically the design got more and more complicated and complex to the point where FDA said, well, this probably isn't waivable at all. And so we appealed and eventually got the product waived. Though FDA is careful to say, well, we're not waiving it based on accuracy. Our accuracy guidance from 2008 is perfectly fine. We still follow that, but we'll come up with a different way to waive your test. This was a test for dry eye disease, not the highest risk test. Important, but not high risk. Well, I left. I went back to my pharma life, did more work, then eventually went to Epstein, where they do a lot of diagnostics work. And this issue with CLIA waivers kept itching at the back of my head. I'm like, I'm going to look at what's going on now. No tests were getting through. This was around 2013. I was like, why aren't tests getting through? I mean, like the old tests, new versions of old tests are getting through, but not the new novel disruptive technologies. And I'm very pro-disruption. And the issue came back to this accuracy requirement. People would go to FDA and they'd say, reference method is not right. You're not 98% accurate. It's okay for moderate complexity labs if you're 90% accurate, but not for waived environments. All these requirements that had nothing to do with the law or CLIA waiver. So I wrote an article and then eventually founded the coalition. And in the process of that, met lots of people. One member of our coalition is the National Coalition of STD Directors, who was trying to they didn't actually make the test, but they're supporting access to a CLIA waived syphilis test, because most clinics that do STD testing don't have a moderate complexity license. They need a simple, easy test to use. There was one that was moderate complexity, but it took 3 years to get through this process of waiver. Ultimately, what convinced FDA was basically outreach from the community saying, hey, we have an epidemic of syphilis in California. I thought syphilis went away like 100 years ago, but it's back. And they needed this test. Same thing happened with fourth-generation HIV testing, which could diagnose HIV 10 to 14 days earlier than the third-generation technologies during the most infectious phase of diseases. I went to some public meetings and met up with folks from ACT UP New York, and they were out there saying, listen, there are these technologies, they need to get waived. There's no reason to hold it to this new standard when it goes to the waived environment. Just need to show comparability. And if we get this in people's hands, we can get people the treatment and change behaviors quickly. If we don't get them in the clinic, they're going to go out, they're going to keep spreading disease. They might not call back ever for the results because it's terrifying to think about what they might say. So, over time, we've had lots of discussions with FDA, and Congress got interested. They put a provision, and I should say lots of other organizations did as well. And they put a provision in 21st Century Cures saying, okay, FDA, I want you to revisit this guidance. And this December 13th or before, FDA is going to release a draft guidance for public comment. And one of the reasons why I was interested in coming here today is because if you're interested in point of care testing, this is really a huge issue. If you're interested in seeing expanded access, or if you have other interest in point of care, it's an important guidance to weigh in on. Probably, I'd say, the most important that's been issued in several years, in my opinion, because ultimately the decisions that FDA makes in terms of its interpretation of law are going to have a tremendous impact on whether we start seeing more CLIA wave tests with newer technologies, more sensitivity, more specificity, and for new analytes, or if we're sort of stuck in this grassroots kind of lobbying effort where public interest groups need to put tremendous pressure on FDA to move on an application. And so that's why I'm here today. I'm sorry I spoke way too long, but thank you for your indulgence.
Okay. So what I'd like to ask all the panelists is what they feel is their primary issues now with companies bringing this new type of innovative technology through the FDA process, and then the second step through maybe even more extensive, the CLIA wave process. Is it the establishment of clinical values? Is it going to be reimbursement? Is it competitive landscape? Is it cost? And we'll start first with Kevin. And if you take a look at your platform, how many analytes do you currently have on your system?
So we have a menu of about 100 off-the-shelf analytes. We also make a homebrew kit, and there's probably been another 120 proteins that have been done via homebrew.
Okay.
So about 220 have been gone through the machine, and we have a menu in neurology that's pretty much seeing in blood brain health, and we've had a lot of focus on this area. Some of the newest biomarkers, really there's no precedent. So many of the pharma companies are trying to get into trials, phase 1, phase 2, phase 3, in an LDT setting with CLIA wave, not CLIA wave, but with CLIA lab. Mm-hmm. And that is probably may lead to an LDT companion diagnostic. Originally, we had an assumption that you had to be an IVD for most pharma biotech, and in the areas of infectious disease and cancer, where we have a lot of menu, we do think that that's going to be more of an IVD companion diagnostic. But in the area of neurology, right now, it looks like this probably is going to evolve into potentially an LDTs, seeing Alzheimer's long before dementia, seeing Parkinson's, ALS, almost every area of neurology, there's now markers that have created incredible correlations in publications in blood. So I think that that's going to be our path for the short term, and we'll talk later about the diagnostic path for some of these areas, but from a companion diagnostic, we think it'll probably be primarily LDT.
So currently, of the 250 molecules, how many have gone through the FDA process?
So the FDA process is an IVD process, and there we are teamed up with bioMérieux, and they themselves are working out, I think, about 4 to 6. And in the RUO market is where the 250 are today. Primarily working with pharma, biotech, and many of them are multiplex now. We have panels of 6 to 8 that we can run on the machinery, but the FDA IVD approval probably will be another couple years from a bioMérieux vantage point from that channel to market.
Okay. So question for you, Neil. Everything today is big data. Everybody uses their mobile phones. Everybody has their information on their iPads. But yet at the same time, in the last 6 months, we've had, it's been all over the papers, a huge number of healthcare systems have been hacked into. they're very concerned about what type of information can be transferred from mobile devices in order to protect PHI. So what has your company done to address this? What have been some of the concerns in trying to use this mobile technology?
Okay, thank you. I think there are a couple of aspects to that. So one of them is about being clear to people exactly what information you are transferring or sending. So clear, upfront, unambiguous privacy statements that tells people exactly what data you're sending and what you plan to do with it in language which you can expect the user to understand. So in one screen, not 96 pages of license agreement where the details are buried away. And then I think the other part of that is about the security of that data once it's collected, so whether it's stored on the phone or whether it's in transit or once it gets to the other end. And I think the parts where it's stored on the phone or the mobile device, that is actually quite straightforward. The mobile operating system ecosystem there makes things relatively straightforward to secure. You can add your own encryption layers on top of that. And the transit, the data in transit part is fairly straightforward to manage as well. Certainly if you're not in China, then you can add virtually any level of security on top of that, and the data in transit's easy. The hard part, and it's where I see the biggest challenge in making a commercial success of any mobile diagnostic application, is what happens to the data once it gets to the other end. So once it gets back to the central server, and how that's secured and managed, and there's a whole ecosystem or infrastructure there to look after and manage. And the reality is that most diagnostic companies at this stage are not well set up to do that. It's not an area that they particularly understand. It involves a whole language and a whole world of different terminology. And so I think that requires a partnership approach with people that really do understand big data and security, and who have a belief in that privacy rather than an objective of how can we extract as much value out of that data commercially as possible, how can we extract as much patient value rather than how can we extract as much financial value.
So the information on your phone is encrypted. I mean, Google can come into our phones and reassort all our pictures and put them in by face recognition and categories simply by being able to allow tracking. So how on that cell phone do you protect PHI information?
Yeah, so I think that requires a two-stage process. The first is to minimize the amount of information you're storing in the first place. So do you actually need that data stored on your phone long-term, or once it's collected and sent back to the central database or electronic health record system, can you delete it off the phone completely? That's the first stage. And then the second is to apply your own level of encryption on top of that. So we would apply encryption layer on top of the basic Google or Apple level of encryption that they provide with the device. And any mobile developer anywhere in the world should have that capability to do that.
Okay, Alidad. So currently, the assays that you're running in your own laboratory, have any of them gone through the FDA process for a clearance or approval? Do you plan on doing that or you plan on keeping it internally within your own company?
So our assays are LDTs, but since we're a clinical lab, we report the results back to physicians, and that impacts patient care. We have gotten CAP accreditation for our laboratory and New York State licensing. And if anybody in this room has been through a New York State licensing procedure, they'll know it's pretty much as tough as it gets, and I'm not throwing a challenge at the FDA to top that. But the first New York State inspection we had a few years ago, we got written up for not having an SOP on how to write SOPs.
Yes, that's correct.
So, when people look, and especially when we talk to people from overseas and they go, do you have an FDA approval for your product? And we say, no, it's an LDT. They sort of look at you as if you're selling some kind of test that has absolutely no oversight or regulation looking at it. When in essence, I've done a few CAP inspections, and they not only look at your total infrastructure of your lab, your HIPAA compliance, your patient privacy issues, they look at the validation data, both analytical validation and clinical validation. So there are processes in place right now, even without direct FDA approval of assays, that look for some level of validation to make sure that what you're claiming as far as the performance of your assay is concerned is actually replicated and documented. So no, we don't have an FDA-approved product. Unless they put a gun to our head, we're not going to ask for it. But we do have CAP-accredited facilities, and our products are all New York State licensed.
Okay, so I have a question for Dan. So you have this nice little PCR device that you can do in 20 minutes that you attach to your cell phone for the data. So in putting these type of systems out into a point-of-care setting, how does your company go about being able to ensure that the testing is done appropriately, that there's going to be proper quality control, which has been a lot of concerns with some of these rapid point-of-care devices. Even data interpretation, how people are going to be able to take that information when it's not a trained laboratorian that may be reading the data.
Well, because our device is a platform that other people develop assays for, it's very much like a cell phone, the iPhone, or any smartphone. The app developer is able to write to our toolkit, so to speak. So we basically are a measurement meter. We are a molecule meter, or what we call a MOM. And it just has the ability to detect the targets that it's looking for, to quantitate, and to do fractional abundance if that's what's necessary, not just molecular diagnostics, but analytes ranging from individual elements, like we have a mercury and lead test, all the way up to full cells, if that's what you're looking to detect. So we don't develop the assays. We just have a mechanism for others to do that. They develop that workflow. They develop what those quality assurances are. So we don't have any control over that. As for the data itself, our device, even though it sounds like a cell phone, it is not a personalized device. It's more like a glucometer. All it does is read data. It doesn't know what the assay is. It doesn't know who it belongs to. All that data is transmitted to the assay developer's cloud. They resolve what that data is, who the patient is, and they send it to a cell phone, if that's the doctor or if that's the patient. Our company's policy is mechanism, not policy. The technology is able to conform to whatever the policy, whatever regulatory processes are in place at the point of use. So if you use that device in the United States, but then you go to China or India or South America or Europe, it will detect where you are and apply the appropriate policy for where it is. So if somebody makes a third-party assay and they get FDA approval or maybe CLIA waiver in the US, but it has not been approved in Europe, when you try to run that test, it'll say it's not authorized. It's also possible to allow the user to run the test and not see the result. For example, liquid biopsy. They can run it at home, but they won't see the result. It'll just say it's succeeded or not. The data will go to the doctor, and the doctor can determine what and when to tell the patient.
Thank you. Well, we'd like to thank everybody for joining us. I'd like to thank the panelists here for today.